So you're now ready as a fully trained MAPS team to decide what project, what modeling project do you want to start. So that means you have to select a story, you have to select a paper or an application of antibodies, and you're going to make a model of an antibody, or maybe the model of the protein the antibody binds. So that's your challenge now, is to decide what do you really want to do. And you should choose anything that is of interest to you. That's the most important thing. But if you're looking for a suggestion, in the last video, we're going to suggest a specific project that we refer to as the Aeronab Story. So why do I like this story so much? First of all, this describes a piece of research that was done by a multidisciplinary group at the University of California, San Francisco. And I like this because, to me, this is just an excellent example of what we refer to as the process of science. You know, how does science get done? Who does the science? Is it collaborative or is it competitive? How does all of this work? My hope is that some of you might be considering a career in the molecular biosciences. So here's a way to sort of get a glimpse into that world. I also like it because it's a story of cutting-edge synthetic biology. And synthetic biology is a fancy new name for a field of research in which people are beginning now to engineer proteins, either natural proteins that are being engineered to do something better than nature evolved them to do, or maybe to change what they do. So this is an excellent example of that. It's going to be a story of how these people were able to screen a library of hundreds of thousands, if not millions, of copies of individual nanobodies, single domain antibodies. And they screened them and identified several that bound to this spike protein. So then they took those nanobodies and they began optimizing them. They began optimizing them so that they not only bound to the spike protein, but they bound better. And remember, this is the whole antibody. This nanobody here is only 1/10, 1/12 actually, of this antibody here. So they have these tiny little nanobodies that can bind even more tightly than this antibody to this spike structure. And they found one. Remember, we said that the receptor binding domain here went back and forth between a inactive down conformation and a active up or open conformation. They found one that bound the receptor binding domain in the down conformation, and that essentially locked it down then into a conformation in which this spike protein is inactive. It's not capable of finding this ACE protein in the surface of a cell. So that's what they did. And they did some other things you're going to read about in the paper that are really clever. So that's the story. And I don't have to tell you much about this story because there's a video and there's a written summary that will show you. We'll give you a link to those in just a minute, and you can discover these things for yourself. But there are a couple things I want to show you. So one of the things I want to show you is on this PowerPoint slide. And this is just a screenshot of the paper that describes this work. And it's not really the paper. It's what we refer to as a preprint. So there is something called the BioArchive Server, where researchers now will upload their paper, the results of their research, before it's been submitted to a journal, before it's been peer-reviewed. So you have to read all of these preprints with a little bit of skepticism because it hasn't gone through peer review yet. No one else in the scientific community has put their stamp of approval on it. But this BioArchive server was put together by some people before the pandemic began, but it has really turned out to be an extremely useful tool for all of these people who are pivoting from their their normal research projects to begin working then on coronavirus projects, and then wanting to share the results of that research with their colleagues in a much shorter time frame than it normally takes to go from submitting a paper, waiting for the peer review, perhaps revising the paper, going back and forth, and then finally seeing your paper appear in a journal. So there's just been an explosion of papers that have been uploaded to this BioArchive server. But I just want to call your attention to this. Here are the authors of the paper. And I want you to count the number of people who contributed to this work. I said it was a large multidisciplinary group, and here's the proof of that. I've highlighted a few names here. Peter Walter down here. I don't know this group at all. I don't really know Peter, except I know of Peter. I know who he is. He's a well-known researcher. Well known, again, for work that's completely unrelated to coronavirus until this paper comes out. He works on something called the unfolded protein response. Completely different. So he, I believe, hope I'm not misstating something, I believe Peter is the senior person in this group. But most of these other people are young people, not too much older than you. So if you're considering a career in the molecular biosciences, here's an example of how people get to become a Peter Walter after a lifetime of doing research. The last author here, Ashish Manglik, you'll actually see both Peter and Ashish in this video. So he's definitely a young person. So I want you to sort of compare the difference, the age spans between people you see here. Another person is Michael Schoof, who is the first author. So this is where everybody wants to be on a paper. I've also highlighted Smrita Sangwan. She actually, you'll learn, she turns out to be the structural biologist who actually determined the structure and deposited the coordinates in the protein data bank that you're going to be using to model a protein. Let me say a little bit more about these authors. I want you to actually read. I want you to sit down with your MAPS team, and I want you to read all of these names. And you're going to have trouble struggling to pronounce some of these names. And the value of doing that is I want you to really understand how international science has become. As Americans, we sometimes have this arrogant view that all the good science is done in the U.S. by Americans. That's not true at all. And the names here is just a testament to the international flavor of science. So one of the great things about being involved in science these days is that you meet people from all around the world in doing that. Okay, I could say some more things. There's a bunch of symbols here. These people are distributed across 17 different organizations, most of them at UCSF, but there are some small biotech companies here. There are a couple clinical groups here, so it truly is a collaborative project of many different people with many different expertises. That's a word. And they all brought this together in a very short period of time. The other remarkable thing is this is work that was done in, I'm guessing, about 6 to 8 months. I'm not exactly sure when this preprint went up. I don't even remember for sure when we found it, but it's really been a very, very rapid, fast-paced thing. All right, so that's the paper, preprint. Take a look at that. I'm not going to say any more because in addition to that preprint, which is right here, there's a video at this link, and it introduces Aeronab. Aeronab stands for aerosolized nanobody. And here's a picture of a nebulizer that is creating this little mist of prophylactic nanobodies. So when you step into an elevator and you suspect someone in the elevator might be shedding coronavirus, you'll be carrying around your nebulizer in your pocket. You'll pull it out. You'll take a whiff of your nebulizer, and that will coat your upper respiratory system with a bunch of nanobodies that if there's a coronavirus that you've just breathed into your system, that nanobody will bind onto the surface of these spike proteins, lock it down into an inactive conformation, and you're protected from infection. So take a look at these terrific resources here, and you'll be well on your way to exploring this research topic.